Hi, everyone. Thanks for joining us for the Boston Epigenetic Society webinar, our November version. I'm going to give just a couple of minutes for people to log in, and we'll get started in just a couple minutes. Thank you so much for joining us. Hello. I want to thank everyone for joining today. We have a great webinar coming up from TruDiagnostic, and I'm going to get started now. I know people are still logging in, but we'll get started. I want to give as much time to the speakers as possible. If you haven't joined us before, welcome. Thank you for joining. The mission of the Boston Epigenetic Society is to promote epigenetics research and to foster connections between academic and industrial epigenetic scientists. And I think today is a great example of that mixing. None of this would be possible without a ton of help and support behind the scenes. I want to thank the leadership team. The leadership team is listed here to the left. They help organize all of these different activities and webinars, and especially I'll give a shout out to John McShane. He's doing a lot of the heavy lifting. And then I want to thank our sponsors. So today, Active Motif is helping us with the webinar and leading the webinar, and this wouldn't be possible without Active Motif. I also want to thank Cantata Biomodal, are also great contributors, and none of this would be possible without them. If you're interested in getting more involved with the Epigenetic Society, there's a few ways to reach us. In our last webinar, we announced our webpage. So we're at bostonepigeneticsociety.com, all one word. And today I'm excited to announce that we have an email listserv. So if you go to our webpage and then scroll to the bottom of the page, you'll see a newsletter sign up. If you go to that on our webpage, you can be alerted to all upcoming events, both in person and virtual. And traditionally, historically, a way to keep in touch is through our LinkedIn page. We also keep everything there. And you can email us at bostonesociety@gmail.com or reach out to any of the leadership team who I just showed you. Today's webinar is being recorded, and it will be available on our website, as are most of our webinars. We're very excited. We have a holiday networking event coming up December 17th. So if you're in the Boston area, come on down, join us. We'll be at the Lamplighter Brewing Company in Cambridge Crossing on December 17th from 6 to 9. And then lastly, we already have our next webinar planned for January 23rd. So I hope you can save the date, and we'll see you in January 23rd when Marissa Bartolome will be giving a webinar for us. So without further ado, at this point, I'm going to turn it over to our MC for today, and John.
It's my pleasure to introduce TruDiagnostics. We have Ryan Smith, who is the founder of TruDiagnostic. TruDiagnostic is a CLIA-certified lab and health data company which specializes in understanding and reporting insights found through epigenetic methylation data. Since 2020, TruDiagnostics created the largest private epigenetic database in the world with over 25,000 patients tested on the EPIC 850K array. From this data set, they have created many new algorithms to interpret methylation data, including algorithms to predict inflammatory markers, disease diagnosis, telomere length, aging, pace, immune cell subsets, cellular replication, and more. TruDiagnostics has also led research in the field with over 30+ clinical trials investigating the best ways to reverse these markers through diet, lifestyle, and prescription interventions. Currently, TruDiagnostic is working with collaborators from Duke, Harvard, Yale, UCSF, Ohio State, and more to uncover new insights from the methylome. TruDiagnostic is also the only commercial company on the market to offer second and third generation aging algorithms, which have shown to be more predictive and precise than the first generation of aging clocks. We also have the pleasure of having Dr. Varun Dwaraka serve as the head of bioinformatics at TruDiagnostics, where he leads the team dedicated to developing and implementing advanced machine learning methods to identify epigenetic biomarkers. These models are crucial for understanding the molecular biology of aging and investigating the associations between aging, disease, and overall mortality risk. Dr. Dwaraka holds a Bachelor of Science in Molecular, Cellular, and Developmental Biology from the University of California, Santa Cruz, and a PhD in Biology from the University of Kentucky. At TruDiagnostic, Varun also serves as the principal investigator leading the scientific investigations conducted by the company with a current H-index of 8 as of August 2024. Many of these studies have garnered significant attention in popular culture, including Forbes, BBC, The Times of London, Newsweek, The Aesthetic Magazine, and Men's Health Magazine. And also on Netflix, You Are What You Eat, and HBO, Curious Case of Natalia Grace, and various popular health and scientific podcasts. His scientific endeavors have earned him a full membership to the prestigious Sigma Chi Xi Scientific Research Honor Society and recognition as a 2023 Foresight Fellow in Biotechnology and Health Expansion, an accolade awarded by the Foresight Institute. So without any further ado, I'd like to introduce Ryan Smith.
Yeah, thank you so much, John, and thanks to the entire team for having us today. We're really excited to be involved in these epigenetic groups. When we first started our company a few years ago, epigenetics was really not even talked about in the clinical realm, and now we're sort of seeing it pop up everywhere and hopefully leading that to some degree. So in today's talk, what we're really going to be going over is TruDiagnostic and what we're doing. We'll talk a little bit first just sort of about the general structure of our business and how it's been sort of growing over these past 4 years. We're still really young, but we've grown a lot over the course of our start in July of 2020. So we talk about the markets that we're hitting, the markets that we're seeing develop, and really where we live, which is translationally. We live in the middle of, I would say, direct consumers, in the middle of physicians who are using this testing, and then also those researchers who are helping us create these better algorithms. So with that being said, we'll sort of hop right into it and talk really mainly about the first problem that we set out to solve with TruDiagnostic. Again, we started in July of 2020, and previous to this business, I had founded a couple other businesses in this sort of cash pay medicine space. These are really made up of a lot of individuals who are really healthy and looking to be very preventative in their own healthcare, and managed by physicians who are helping them do that, but who are collecting some of the most amazing biomarkers that you can imagine. Everything from the full multiomic suite of proteomics and metabolomics in full genome, but also a lot of really great interventional data. And really, this whole market is motivated out of one big thing, which I think is sort of the first product we created at TruDiagnostic. And so I always like to start these presentations by showing the top 3 causes of death in 2020. This is obviously the year that we were founded, and then showing really the top 4 relative risk factors for every single one of those: heart disease, cancer, and COVID-19. And as you can see, the 4th risk factor here is much, much higher than all the others. And so some of the risk factors you'd encounter here are things like smoking or obesity. But there's one that really is around 30 to 40% of the relative risk for each of these conditions. And we always like to draw attention to what that is, which is generally aging, and particularly biological aging. It dwarfs the relative risk factors of things like smoking, obesity, and hypertension, despite how much everyone knows these things to be incredibly important to disease risk. And so one of the first things that we did at TruDiagnostic, the first product that we ever launched to our consumers, and our consumers being those healthcare providers, was diagnostics for aging. And this market has been growing, and we've been lucky to grow with it. The longevity market is estimated to be over 4.7 trillion. And even just adding 1 year of health span for an individual would have the economic impact globally of $37 trillion. And so this is a huge area of research now now that is growing. And we really aim to do this by quantifying this biological aging process. And so again, we all know people in their 60s who look like they're 50 and people in their 50s who look like they're 70, right? And so we know that there's a process here that's not just related to how many times the Earth has taken a trip around the sun since we've been alive. And we really want to measure that molecularly. And so we've been doing that with DNA methylation. You've probably seen, thousands of publications on these DNA methylation clocks since they first started with Steve Horvath's work in 2013. But we've really tried to improve these and make them better so that our physicians can actually use them. And the way that they're using them is by monitoring this aging process and hopefully reversing this aging process. Later in this presentation, Varun will go into some of the ways that we found to do that, not to bury the lead. There's some amazing ways to measure this, but there's also some amazing interventions that we're now just starting to validate. And we're really hoping that aging will start to be viewed as a risk factor, which is both manageable, but also measurable. And that's really what we're trying to do. So we've done this over the course of these years by improving these clocks and how we actually measure this biological aging process. At the end of the day, what we're really trying to do is to predict outcomes better than anyone else. And if aging is your biggest risk factor, aging should be able to predict those other outcomes of disease very, very well. And so, as you all know, this started in 2013 with these biological age clocks from Gregory Hannum and Steve Horvath. These were first generation chronologically trained clocks, which were just meant to estimate someone's age. But in really 2017 and 18, this progressed to second generation clocks that were trained on biological phenotypes of aging. And these clocks were much, much more predictive of outcomes. Since Morgan Levine's PhenoAge in 2017 and beyond, these clocks have gotten incredibly advanced. And we really now feel like they're finally able to be used in clinical settings to offer actionable information and really for us to quantify who is at the highest risk. And so Varun will go into some of the work that we've done into these areas, but this is the market we set out to meet first. And we've done this through collaborating a lot with researchers, but also through that physician market. When we started this business, we had over 40,000 cash pay healthcare providers within our network, and they've been really the people that have been implementing our testing with a high degree of scientific knowledge and resolution so that they can use these in the way that they're meant to be while we continue to perfect them. But this is what we do. And so you can give an idea of some of the algorithms that we've helped create or license. This is a full list. We've done the Dunedin Pace, which is the only third generation clock from in collaboration with Duke and Columbia. We've done Omic Age with Harvard, Symphony Age with Yale. These are some of the best clocks that are out there now. And now they don't just provide an overall age, but they provide sort of a breakdown of why you might be aging. And this is extremely important to our physicians so they can sort of manage the biggest risk factor of all time, which is that biological aging process. And so we've sort of created this market. We probably now, I would say, have over 90% of the biological age testing market here within the US and are expanding internationally where we can offer these things for clinicians and patients to use. In addition to our healthcare provider network, we do also have a very small direct-to-consumer market, which is led by people like Brian Johnson, who we've created the Rejuvenation Olympics Aging with. For those of you who might not be as familiar with this aging industry, Brian is a serial entrepreneur who's had a lot of success, who is now planning on or trying to live forever and using some of these tools to tell us what's working and what's not. And so now we have even competitive leaderboards to tell us who is aging the best. And so we've sort of stepped into this biological aging market with creating new and better tools to both, I would say, measure this process and then manage care going forward for all of our physicians. And so that's been really our major area of focus and research. But our bigger vision of our company is to really do as much as we can with epigenetic methylation data. We have built the largest database to date of DNA methylation data, and that's about 125,000 patients now. So it's still bigger than the US Veterans Health Study or the Generation Scotland cohorts. It is a massive amount of information that we've been able to collect and generate from our commercial markets. And so our company's grown pretty expansively. Where we have a lab here where we do most of this methylation work. To date, most of that methylation work has mainly been on the Illumina array platform. platforms just for scale and price, but I think we're really interested in branching out to other methods of sequencing to get things like the 5-methyl hydroxymethylcytosines and getting better sequencing related data. But this is our vision, is really to own this platform of analysis as much as possible. And to do that, we need to generate these large multi-omic and phenotypic data sets. And we've been able to do that with our physicians. So we're obviously committed to research, as you know. Now we have over, again, hundreds of thousands in our data set. We've published over 24 studies. I think we'll have probably 5 preprints in the next 3 weeks, and collaborate with tons of different universities. And if you don't believe me, you can you don't have to take my word for it. I'll turn it over to the words of our best scientific collaborator.
This is the best test I've ever taken in my life.
This was so cool.
Yeah.
Yeah, so obviously these are the Kardashians. Even though we do a lot of research, we also do a lot of pop culture. As you might have heard in that introduction, we've been on HBO, Netflix, the Kardashians, because people are really interested in this topic of epigenetics, age, but I think they're also interested in epigenetics just generally, and that's a market we've been able to hit. With some of our new tools, we'll be able to do a lot of different things, and I think that outside of giving you the introduction to TruDiagnostic, I also want to talk about some of the applications. Again, we want to create the Rosetta Stone for epigenetic methylation, learning how to read these methylation patterns to inform us about your sort of health status. And we're doing that as best we can. Just to give you an idea of the current applications of DNA methylation, and I'm sure many of you on the call are familiar with this, but we already have ways to be prognostic about different diseases. Are you likely to develop side effects of a certain medication? Are you have a 5-year, 10-year survival line? What subtype of diabetes might you have? We can now even subtype diseases into further resolution, which then handle both what drugs we might recommend or what outcomes we might fear. We can now diagnose disease for methods like schizophrenia or bipolar or PTSD, things that traditionally only had clinical diagnosis and not molecular. Now we can, again, turn this into drug discovery methods and help create new product. There are now multiple companies out there who are doing epigenetic therapies, and we can use all of this data to help guide the area where those are best. And obviously, even already in life insurance and forensics, people are using these DNA methylation clocks to both predict death, as well as to see how old you are if you're a refugee or into a new country, or if you are essentially leave DNA at a crime scene, we can tell you how old you are. And this is just the beginning of all of the applications. So in addition to what we do in our TruDiagnostic commercial testing to physicians and direct consumers, we also do a lot more with DNA methylation. We have a lot of B2B partnerships here within our lab where we're bringing different arrays to market, so we can actually design the arrays or the different analytical tools to measure methylation. We also have gone through just different approval formats to help get methylation specific tests ready for the FDA for things like postpartum depression. We do a lot of research sample processing here, both for universities as well as third-party systems. And then obviously we've created a whole platform to bring laboratory developed tests to our clinician market. And Varun will talk about some of the new tools that we've created here. We also have a DNA methylation analysis suite, which uses all of our algorithms and automates all this analysis to make it very, very easy. So that's just an overview of our business from top to bottom. Again, I think that one of the things that's important to mention is just the data that we're collecting and the positive feedback loop that this data creates. Because, when we first started, we had really one algorithm, which was epigenetic age, and a chronologically trained version at that. But over the course of our 4 years, now we have exponentially more algorithms. And the way that we've done that is by collecting data and using our patient and physician data sets to create some really amazing algorithms. So for instance, now we can tell you your immune cell subsets with less than a 3% variance versus flow cytometry. That's a $6 billion industry, and we just add it as a feature. We're able to tell you micronutrient testing, your omega-3 fatty acid levels, your hormone levels, your B vitamins, all through the same data that we were using to tell you your biological age. And we've done this just through, again, creating more algorithms, which help report more feedback to our physicians. Those physicians use the test more often and then create us more data. And so this positive feedback loop is why we're so excited, is because the pace of development at TruDiagnostic has exponentially increased, and we are super excited about where the future is taking us. So in the future, we do imagine that we will have one test to predict many. I didn't wear my black turtleneck to look like Elizabeth Holmes here, but we really do think that DNA methylation can capture a lot of the data that we would want to report on for clinical use. And we have a lot of really good updates here that Varun will probably talk about on why we're so excited about this. So this is sort of where we're thinking about going, but I also want to talk about why we're so excited about it. And that really comes down to this idea of clinical utility. And for most of the people here who are working in epigenetics, this is probably not a surprise, but one of the big limitations both for insurance companies who fund genomic work is that they really have to have some type of clinical utility. The physician has to be able to use that data in a way that manages care or changes the patient. And for polygenic risk scores, this has been really, really tough to document, because do interventions actually change that risk outside of things like gene therapy? But in the case of these epigenomics, this is really where we're focusing. We're really trying to deliver tools that have a lot of clinical utility for the patient, comparing them to things like polygenic risk scores. These things are modifiable, changeable. Not only do they improve how the physician can use this data, but they also avoid, I would say, companies like 23andMe having less repeat customers and therefore not being in a state that is financially lucrative. And so it's good for the labs, it's good for the physicians, it's good for the patients, and we're super excited to bring this to market. But to date, we've also realized that it's been really hard to bring these things to market. And that's because of the tools and things that are being used in the research area now. And so before I hand it over to Varun, just lastly, I want to talk about the tools that are being used and where we think there's the biggest opportunity for anyone else in this space. And if anyone is working in these areas, we encourage you all to reach out for us for collaboration and partnership, because we do a lot of this and now honestly have more data than we can handle. So when we talk about what's common now, we've seen an explosion in epigenetics, but particularly just in EWAS studies. So here you can see, the growth from 2010, 2023 of just EWAS studies that are conducted per year. And we've done a lot of these at TruDiagnostic, and are treating them much like these genome-wide association studies. But we've also automated software to be able to do this. So we can look at, for instance, hyperbaric as a therapy. We've published on cannabis use and smoking status and alcohol signatures. We have more epigenome-wide association studies from even things like medications to disease to lifestyle effects to even zip code based on pollution markers or environmental exposures. And so these EWAS studies have been conducted at a really high rate, and they've taught us some really great things. So for instance, we know that there are certain CPGs that can be used to create these smoking signals. And now even our prediction of smoking status is even better and more related to disease than self-reported smoking status. So what we're reading molecularly is even better than sort of your own knowledge of your own behaviors. And we can even track how these change with different behaviors. So here you can see how methylation is decreased at a certain CG just from data within our first 3,000 patients, which looks pretty conclusive with massive significant p-values. And so this work has been done a lot. It's being done on what we do on a day-to-day basis. But unfortunately, it tends to die here. Some people use this information to project biological significance using things like this great software from Stanford that can tell you about the biological, cellular, and molecular significance. But this needs to go further, I think, for clinical application. And we've been really disappointed that most of these analysis really stop here. And so we want to help create ways to continue that analysis and turn the insights that we might find from these EWAS studies into usable tools that can then impact the clinician and patients. And so we built a DNA methylation analysis suite software to do that. This goes through all the different sort of bioinformatics steps you'd have to do from start to finish, which can be complicated. But at the end of the day, I think that this is really to set up Varun to talk about how we're actually turning these methylation data into actionable information. So how are we taking all the data that we're creating to generate algorithms, to validate and test them, and then ultimately roll them out where they can be used? And this translational area is really fundamental to the identity of our company. And so this is sort of what we're doing, but we know that in order to make it applicable, we must train algorithms. So, using things like XGBoost or penalized regression or some now language learning models or variable autoencoders to create really great clinical tools. And so we're excited to really now take this next step to get way beyond just EWAS studies and association studies and get into things where we can create causal, dynamic, and changeable algorithms that can be used by patients and physicians. So with that, I will go ahead and turn it over really quickly to Varun to talk about some of the specific applications and get into the science.
Thank you. Yeah, thank you, Ryan. Hopefully my slideshow is now sharing. Yeah, thank you, Ryan. That was a really good way to set everything up. And I think what my role here today will be is to kind of maybe unveil some of the ways that we're actually leveraging a lot of this epigenetic aging signatures. And so kind of my segment is going to be the applications of these epigenetic aging clocks and then methylation risk scores using whole genome methylation profiling. And so I don't want to harp on this anymore because I think Ryan did a fantastic job setting this up. But I do want to recognize that given the environmental and exposure effects upon DNA methylation, you can actually look at a lot of variables such as methylation QTL variation, age dependencies, cell type specificity, exposure dependencies, and outcome associated metrics. And all of this can be captured by DNA methylation, and that is essentially what we are leveraging when we think about how do we develop predictive models. And so as a brief framework, how are we collecting a lot of these samples? As Ryan said, we do have a healthcare practitioner and direct to consumer market, but even with researchers who just want to have sample processing, what we are able to do is, especially with the HCP and DTC, we are able to collect a lot of the blood using dry blood spot cards. We go through using the Illumina Epic V2 custom array, which we can speak about a little bit more later, and undergo our entire bioinformatics pipeline where we do QA/QC checks and build out not only the beta values that are associated with the methylation values, but also we take in metadata. And that metadata is very important because that is actually what we're using as a test data set to really validate a lot of the algorithms that we build. And so, as kind of a description, whenever we're building these algorithms, the best way to kind of think of it is looking at it as a funnel. The machine learning process will be funneling down all of the CPGs, any type of methylation surrogate that's available, and potentially some demographic information. At a minimum, you're looking at sex and age, and pretty much training this using some type of machine learning or AI methodology to whatever you're interested in. And so using this kind of framework, we can actually train or select CPGs and features that really come about to building a lot of these methylation scores or predictors. And just as I kind of alluded to, AI and machine learning is really at the centerpiece of creating a lot of this information. So things like methylation risk scores, exposome prediction, immune cell subset prediction, mitotic clock, telomere estimation, all of these conduits are really held within the ideology of AI and ML and using AI and ML here. And so I'll describe maybe 3 overall use cases, even though, as Ryan said, there are many use cases, but the 3 that we're really excited about are kind of the first one, which is estimating biological age. I'm going to recall this slide mainly because the landscape of the clocks, there are a lot. And so you might be wondering, why did we go about creating our own predictor, which is a foreshadowing of what I'll be talking about, when there's so many available already? Well, it's the context of why are we wanting these predictors? And so even with the first generation, we can think of these as different generation where they're trained to chronological age, or even second generation where you're trained to a biological score, or third generation where you're essentially looking at a model that is tracked longitudinally. Why didn't we just go this route? Actually, we kind of did. We selected a few based on what the end user really wanted. So for example, I'll talk about 2 that we've actually been able to license. One is called Symphony Age, where we can actually use blood as a surrogate to estimate age from other tissue types. And the reason why this actually kind of works is because blood is a circulatory tissue and it can capture signals from across the organs. And so in this preprint by Raghav Sehgal, who actually developed systems age from Albert Higgins Chen's lab and Morgan Levine, what they were able to show is that that signature of that tissue-specific age is actually very well captured in blood, and the identification of those CPG sites allows us to now take a blood test and actually understand what those individual tissue types, what their ages could potentially be. And so in the context of how we deliver this to, let's say, consumers, we are able to actually say not only from your blood, we can actually estimate what your overall symphony age is as a composite of those individual tissue types, but also your lung age, metabolic age, musculoskeletal age, and so on and so forth. And what is very important here is the idea that we can now have finer resolution. You're not just an age based on your entire body, but now you can actually look at which organ systems or organ functions are actually aging at a faster rate, and maybe allow for a more personalized methodology to really attack that. The second clock that we really looked at was the pace of aging. And the reason why we really looked at this was because of the fact that it was longitudinally trained. You're following this individual cohort from Dunedin, New Zealand, from the time of birth in 1972 all the way until 2019. What they were able to do in their first paper by Daniel Belsky in 2015 was create this pace of aging score based off of 19 different, I believe 19 different covariates, clinical values. And then so they were able to create that pace of aging. But in order to make this into a DNA methylation test, what they ended up doing was actually training, similar to how I kind of set up that framework, using the pace of aging as a training feature that you're going towards, or the training output. You're picking out CPGs that are predictive of that pace of aging just using DNA methylation. Now, this is great because what it is essentially a pace of aging score, where if you're above 1, you're aging at a faster rate. If you're below 1, you're aging at a slower rate per every year that you pass. But what was really exciting about this, and how we also look into picking specific models, if you will, is the association and responsiveness to treatment. And so there were multiple studies, kind of what Ryan was mentioning. We even took part in one of the studies where we actually started to see where Dunedin pace was actually responding to, and showed that PTSD, so individuals with higher PTSD and depression show higher Dunedin pace. Individuals that were potentially on a vegan diet showed lower Dunedin pace compared to their omnivore counterparts. And fun fact, that was done actually in a set of twins. And things that we already know decrease biological age. For example, lower caloric restriction showed a lower Dunedin pace. And so using this kind of a framework, we were a lot more confident that picking this clock was the right way to go, essentially. However, we still wanted to kind of take this to the next level, because now when it comes to aging, DNA methylation, we acknowledge, is not the only facet of the molecular biology that contributes to the aging patterns. And so what we wanted to do was maybe take this from a holistic molecular biology perspective, by incorporating proteomics, metabolomics, methylation data, and including the clinical data. And so what we did was, in this paper, in this preprint called Omic MH, is that we developed an approach to integrate the multiple layers of aging biology. And by integrating all of that, we then, similar to Dunedin pace, trained it back to DNA methylation. Because again, as a company, what we are doing is processing a lot of the 850K data. But we want to be able to leverage that 850K data in a way to maybe have much more of the signals coming from the proteins, metabolites, and clinical values as well. Now, that paper is pretty hefty. I would highly recommend maybe looking through that. But some of the reasons why we were very excited about this is because, again, age is a representation of disease risk. And what we care about in terms of being able to provide information is not necessarily the age of the individual, but what does that age mean in relation to the overall disease risk that that person might face. And so overall, what we started to see is, when we compared this in the Mass General Brigham cohort, what we started to see is that it shows significant association to disease risk and all-cause mortality, suggesting that this epigenetic clock is biologically helpful and useful. It also showed better prediction of overall survival over 5 and 10 years, as shown by these ROC curves. And what you'll notice is that the AUCs of the specific clock, for example, Omic MH, is shown right here compared to a couple other contenders, including just your chronological age, which shows a 0.78. So even compared to chronological age, Omic MH is doing a better job at predicting whether that individual will survive in 5 years or not. And this is all great as a validation, but how do you bring this to the customer? And so this is an example of a report where you can now, what we can actually do is not only provide an Omic MH, but compare that to the calendar age of the individual. But again, what this age is essentially, you can think of it as a age score. And so what's really useful is because of the fact that we've expanded upon our data set and now have about 35,000 individuals that are representative of a relatively healthy population, we can then look at the percentile of that individual in relation to everyone else. And so that way we can actually start to see, are you really at an elevated, is your age really at an elevated percentile or not? And then also track them as long as those individuals are taking repeat longitudinal tests. But because we actually trained this with mortality in mind, we can also estimate the risk of disease based on all the diseases that I showed in the past slide that we collected from Mass General. We can actually estimate using Cox proportional hazard models that we modeled to all-cause mortality, cancer, stroke, type 2 diabetes, and so on and so forth, all based on this omic MH value that you're gathering. So this really provides a lot more insight into not only what that age means, but how that relates to your overall risk of disease. Now, to shift gears, we went through a little bit of a, let's say, soul searching, if you will, with our first use case, because what we start to realize is that we can go crazy and start to see what the relationship with methylation is to all of these other proteomics, the metabolomics, and the clinical values, because sometimes for the clinician, the age itself may not be that discernible, but let's say if you can impute CRP, that actually makes a lot more sense to a clinician because of its association with, let's say, inflammation and all that. And actually, in a paper from Steve Horvath's lab, what they actually showed is that if you use DNA methylation as a surrogate of, let's say, HbA1c, what you can do is you can actually effectively model it, but it provides a benefit because that DNA methylation-based biomarker, instead of the plasma biomarker, the analog, actually, the DNA methylation biomarker represents a longer average estimate of the biomarker concentration. So this got us thinking about creating these epigenetic biomarker proxies, or these EBPs, which are essentially DNA methylation surrogates of their phenotypic clinical protein and metabolite output. Now, we're not the first people to do it. Obviously, Steve Horvath's group has been looking into this, especially with the inclusion of Grimage. Also, Ricardo Marioni has done this. But what we have been trying to do is actually use the multi-omics to greatly expand upon these EBPs, and kind of using the same framework of machine learning to identify which CPGs are actually effectively able to model that biomarker. And so in this preprint that we will hopefully put out in the next week, what we actually did was we leveraged DNA methylation to create these EBPs, going from approximately about 3,000 different models, and really looking at which models showed biological and clinical utility, especially for the association to disease. So in this flowchart here, the main thing to really consider is the amount of time and the amount of modeling that it took to get down to this set of 1,694 approximately models. We used both ElasticNet, but also XGBoost, which has not been readily used in the field. And the reason why we used XGBoost is because, especially when it comes to modeling other types of proteins and metabolites, realizing that there's a linear relationship may not be the best realization. And so we ended up using more of a gradient boosted method, and also using transformer-based learning to really let the data speak for what the best model is. And so we filtered this based on a correlation between the observed values that we were getting from all of these multi-omics to the DNA methylation estimate. And we essentially retained everything that had a correlation of 0.2 or more. But what is very important, again, is not the value itself, but its association to the disease. And so one use case of this is that what we start to realize is that these EBPs can be then used specifically as a panel for particular diseases. And so what you're seeing in this heat map right here is essentially which of these particular EBPs are associated with which different cancer types. And also bear in mind that these were also selected to make sure that these EBPs showed a consistent association to the disease as their actual observed counterpart. They also perform similarly longitudinally. So even though at a specific point, they might have some variation, what is really striking is that these EBPs actually show similar longitudinal changes as their counterpart. And so, for example, EBP for neutrophil percentage, especially with the individual 9, shows a very similar story in terms of the longitudinal tracking compared to something like even vitamin D, where even though there are deviations, but for example, individual 9, that longitudinal tracking is consistent. And in this paper, we will also be discussing a little bit of why those deviations are, because very similar to what Steve Horvath was talking about, what we're starting to notice is that these deviations actually represent a more stable biomarker from a day-to-day perspective compared to the variability that you might expect in a plasma biomarker. And what is important, though, is that the majority of these EBPs outperform their counterpart in disease and mortality association, which is what we're very interested in. You don't have to worry about all of these numbers, but the main thing to really look at is the percentage of similarity to hazard ratio and odds ratio between those EBPs. And so approximately 62% or 64% of the ones that we identified actually showed a consistent association to disease and overall mortality. Again, the details are very important and they will be available in this preprint. One other aspect of what we do is that we also test these predictors readily. And so we've actually used a few of these EBPs in different types of interventional studies. And so, for example, in this study of looking at ketamine usage amongst individuals that were pre-diagnosed with PTSD and mild depressive disorder, what we were identifying were actually EBPs that made sense. 3-beta hydroxy 5-cholinate has been shown previously to be to actually start increasing when individuals that were treated for depression with ketamines were actually showing better responses. And what we're showing here is that the EBPs actually track consistently with what has been previously seen. Now, this is fine. But you have to remember, we have 1,694. So imagine how many other additional associations that you can find that have never been considered before. And so it's really an effective discovery tool, especially in the research setting. But even as a test, you can implement this. For example, in this biohacks eval that we're associated with, what you can do is collect a blood sample, run through the DNA methylation pipeline, and then essentially look at your DNA methylation biomarker status, and then see if you're in the early 10th, let's say the bottom 10%, the top 10%, or in the middle, and then also your associations to age. And so that's kind of what we've been looking into and what we will be releasing in a new product from TruDiagnostic called TruHealth, where we're actually taking a subset of these models and actually being able to offer them as a test, looking at different aspects of nutrition to let's say the effect on different changes of your body. Essentially, there's so many more that you can look into. Nutrition was one that was really interesting there. And as I mentioned, this becomes a cost-effective screening tool because now you're taking one single blood-based sample collection, running it through the Epic, and now getting estimates for about 1,000. It's more than 1,600 biomarkers. And if you consider the cost of doing that individually, you're looking at almost $1 million, close to $1 million, versus a test that is a couple $100. Last but not least, the last case that I want to mention is the usage of estimating not age-related disease, but methylation risk scores. And so, for example, when we do a lot of this work with epigenetic clocks, what we're doing is that we're using the epigenetic clock or the EBP as a surrogate to estimate that risk of disease. And quite honestly, as you might be already thinking, why not just skip the middleman and go directly to the disease itself? And so we've had some early work. We're starting these modeling of methylation risk scores. And so what we've been seeing is that the AUCs, or the area under the curves for these models, or the ability to predict, have been relatively high. And while we can do this for type 2 diabetes, COPD, stroke, the interesting ones and the ones that we're really excited about are things like congestive heart failure, depression, and even chronic kidney disease. For example, with depression, there's really no real molecular test that's available to really look at the diagnosis of, or look at the risk of depression. But versus one that we created just based on DNA methylation, we have an AUC of 0.711, which is highly predictive. In the case of chronic kidney disease, we're blowing out previous contenders that had an AUC of 0.71 to approximately 0.9. And so as we move forward, what we're going to be doing is we're also going to be entering this area of methylation risk scores, mainly because DNA methylation from the capturing of the signals that are environmentally driven seem to have a better estimate of the disease risk based on that use of that biomarker. And so in summary, there are really 5 points I just want to harp on. To impact aging, we need diagnostics and treatment. Epigenetic clocks initially serve in this capacity. With the current epigenetic clocks that are able to estimate epigenetic age, which are clinically relevant and predictive of disease and health, and that's where we kind of come in to really assess which ones to use and bring that to market. But more importantly, we can also then get resolution to individual organ systems using symphony age or the current pace of aging versus overall age. So now not only are we able to get a holistic number, we can actually segment based on immediate pace of aging or the individual organ level based resolution. This whole concept of epigenetic biomarker proxies now provide even more resolution from DNA methylation data. Again, this is the same DNA methylation data that's being used for the epigenetic clocks. Now you can get use that same data set to now infer those more than 1,600 biomarkers. And this is a low cost, easy to collect method, and it's coming directly from that same data. And what we hope is that now moving forward, especially in the next early Q1, we will be developing methylation risk scores to directly estimate age-related disease risk without actually looking at the middleman, the epigenetic age and the EBPs. Now, we'll still calculate those, but what is that overall disease risk if you didn't have to use that conduit? And so with that said, I think Brian did a little bit of summary, but I do want to recognize everyone at TruDiagnostic. It's a huge endeavor here, and we generally like each other. So I wanted to highlight the corporate team, and then also our significant collaborators at Harvard Medical and Mass General. So thank you.
Hey, Varun, Brian. Ryan, sorry. Thank you very much for a great talk. I'm still blown away by what you're able to do, especially the use case too, the EPPs. I was always wondering how can you use that information? Aging, obviously super interesting, and it's a hype right now, but really using EPPs and the correlation with the biomarkers are even better than the conventional biomarkers. I think there's a huge potential. I'm a cancer researcher, and I was always waiting for something like that. I quickly check if you have questions, but maybe before, maybe I can have the first one. An obvious one is now you measure the 5MC, right, with the Illumina chips, and as we know that method cannot discriminate between 5-HMC and other oxidation states of DNA methylation. Maybe if you can give us an outlook for where this is going, your plan on maybe differentiating the different marks on DNA. It leads into the omics question, I think, yeah. Not sure who
Brian, do you want to take that or?
Yeah, well, you go ahead. But particularly
Okay.
leading to the omic data, yeah.
Yeah. So we recognize that 5-hydroxy methylation is also another biomarker, which especially at ASHG when I met John, I think Biomodal showed a little bit of the usage in methylation risk scores. One area that especially I've been thinking about, and this might be an area that we go into, is using this as a screening tool, using the epic arrays as a screening tool, but then maybe potentially going into something like a 5-methyl hydroxy as a way to really look at individual methylation risk scores. So you might have something like you run through this, you have a panel, running an epic array is very cost effective and seems to be bringing out information that's very applicable. But then once you get a positive hit, how do you go from there? And so maybe the development of additional methods using 5-hydroxymethyl or some other type of omic data could be then leveraged from then on. So I think it could be used in tandem, but at least from the methylation array, it is a very cost effective and as much as I would say encompassing, I do recognize it only covers about a percent of, 1 million out of the 28 million CPGs. it appears that even based on the AUCs and associations to disease, we are getting a ton of signal just as it is. So we'll still use it as a way to incorporate additional algorithms.
Okay, that's great.
And
sorry. Well, yeah, no, sorry. I was just going to extend on that into the multi-omics question, I think, that you were asking there as well, Michael, which generally, I saw in the chat is sort of asking about how we integrate all of these omics. And for us, we have decided to integrate them all through the lens of the epigenetics, which is why we've created those epigenetic biomarker proxies. As much as we love multi-omic investigations, they're obviously very, very expensive. And they also require a lot of things that can be prohibitive in research and study environments, like plasma draws or phlebotomy, whereas with the DNA methylation, we just need a couple drops of even a finger stick blood card. And so in the multi-omics analysis, we certainly have yet to integrate them into a single model. What we're really doing is trying to train methylation to incorporate signals of those omics together.
Great. Thanks for answering this question. I was about to ask it from the chat. Maybe back to the clinical trials. You mentioned in the intro, right, that the potential and the EPPs clearly, right, with the correlation with the biomarkers looks very promising. I'm very eager to see your bioarchive paper. But can you comment on where if there's any clinical trial who tried to use DNA methylation as a biomarker already? Have people done that to predict response rates already? I'm not sure where the field is. Yeah.
Yes. So Varun, I might go ahead and give a start here. Yeah, the epigenetic biomarker proxies have not necessarily been used to date to measure efficacy of a certain intervention. However, that will change with a lot of studies that we currently have in publication. And so we'll be doing, for instance, interventional trials on things like semaglutide, on statins, where we show significant decrease and sort of a matched decrease among the observed biomarkers, as well as the epigenetic biomarker proxies for a host of things like cholesterol and lipids in the statin trial, and then obviously a lot of blood sugar and inflammatory markers within the statin or the GLP-1 based analysis. And so we are certainly showing that now. The one interesting and thing I just want to remark on that Varun mentioned as well is that we do imagine that these have benefit over their classical biomarkers. And 64% of the analysis that we did, those epigenetic biomarker proxies had higher hazard ratios or odds ratios than their matched observed surrogate from a plasma analysis or a proteomic or clinical or metabolomic analysis. And that's why we're so excited. It does look like we're getting a sort of a longer running average, and that can inform us much more about the chronic disease, maybe less so on the acute, though. I think it might be pushing us more into a direction of long-term stable changes and less into acute clinical care settings.
Great, thank you. Yeah, it's amazing, super promising. We have a question from Susana Gonzalez. Or Varun, you wanted to comment on the previous question?
Yeah, actually, I wanted to put in a plug. So as a company, we also have very good ties with academia. So if you have an intervention you want to try, hit us up.
Yeah, that's the place to ask for it. Definitely. Great. Yeah, Susana Gonzalez is asking, do you take into account the circadian oscillatory patterns and epigenetic modifications? Right? How is this in your models reflected? Yeah.
Yeah, so I will say, Susanna, one of the things when we did this initial omic image is that we did not filter per se from the CPGs that are associated with oscillation, circadian rhythm. So it is quite known that from the generation 1 clocks that there is a oscillation in terms of like the time of day that you collect your piece, like let's say your Horvath age and all that. However, the usage of principal components has also been very helpful to kind of mitigate some of that, because essentially there's a sect of principal component clocks that were built. What they have been able to do is effectively, rather than look at individual CPG sites and weight them individually, you're taking a conglomerate, a set of CPGs, building that into a principal component, and then using the principal component as a way to estimate the overall age. And so if you have, let's say, a circadian rhythm associated CPG, it essentially gets drowned out by other CPGs that may not be circadian rhythm based. And so that way you're able to modulate it that way as well, or correct for that modulation. As we move forward, this is one of the things that we will be taking into consideration. There is a paper coming out which looks at the time of day as an effect of these epigenetic ages. I will say the preprint will be out soon. One teaser I can provide is that with the EBPs, we're not seeing that oscillation from time of day, which is actually really goes back to what Steve Horvath was saying in that paper, where it might actually be a longer running average. And because of that longer running average, the oscillation is not really, let's say, affected as much. And so, yeah, be on the lookout for that. And if you want a early access, please let us know.
And to add to that as well, Varun, I did answer that question in the chat too, just to sort of talk about, there are studies out there which describe it, which we're doing follow-ups on. And interestingly enough, we found not only circadian rhythm variations, but seasonal variations as well. And even I would say some associations to things like daylight savings, for instance. And so we're really excited to publish a follow-up there, but it will probably be a few months before that's out.
Yeah, I still hope they stop this data like changing. Okay, just quick reminder for everyone, right? There's in Zoom, there's a button in the middle, Q&A button where you can type your questions if you have some. We have a question from Vas Agrawal, right? Who says, thank you for the talk. He's asking about the topic, cause versus consequence of these epigenetic markers, right? Is basically DNA methylation causing aging, or it's just a marker? You want to take that one, yeah?
Yeah, so I'll go ahead and talk about maybe some of the two questions left. Just again, I know we're running out a little bit out of time, but the first one I think is the correlation versus causation sort of question I see there in the chat. If that's good to speculate on, I certainly want to do it. Which is that generally the method for developing causal algorithms with DNA methylation has been involved using Mendelian randomization, and the Gladyshev lab from Harvard did come out with some Mendelian randomization based clocks called cause age or adapt age. And we think that was a step in the right direction, but unfortunately, the hazard ratios for predicting outcomes of that still were a little bit limited. And so we're hoping to take some other fun approaches to make these more causative versus just correlative, including some W function Lambert analysis of multiple longitudinal draws. We now have over 1,500 individuals in our data set who've taken this test 3 or more times. So sort of following them out to establish causal effects with some equivalents of those PCAs that Varun was mentioning, but in longitudinal data. So we're really excited about that. But we do know that you can establish causality through methylation data. You just might need some other data there, and that not just in aging, but in almost any disease-specific category. And so super excited about where that's going. And for us, are certainly shifting a little bit more away from just aging and into disease-related metrics that can be used clinically. In addition to that, Genevieve, to sort of talk about bipolar and schizophrenia. Yes,
maybe we can quickly state the question for the audience, right? Genevieve was a speaker a couple of months ago this year, right? And she's asking, right, if she understood it right, and I have the same question, right? If you can really differentiate between bipolar disorders and MDD and schizophrenia, yeah. And that would be
definitely. And so the answer is that I don't. Yeah, I don't know that we can necessarily differentiate, but we, have algorithms now that can, diagnose schizophrenia with a 98% area under the curve. For our depression risk predictors, they're not quite as good. I think we have an area over the curve of 0.82 or something. But yes, these things can absolutely be done with just molecular signatures already. We're about to release a paper. Actually, the preprint might already be out on looking at bipolar specifically and the effects of certain bipolar medications on aging, which I think is also very interesting to mention that almost every psychological condition is also related to changes in DNA methylation aging, which is very interesting as well.
Great, thank you. We have a question from Bartholomew Diouf. Sorry if I butcher the name, right? The question is, did you look at telomeres, right? Repetitive sequences, and is this possible with this technology?
Yeah, so one of the things that I kind of mentioned in the AI segment was that you can actually use DNA methylation as a way to estimate your telomere length. And so in a lot of these interventional studies, especially the ones that we've been affiliated with, we do look at DNA MTL, and especially with the original paper that described this, which was Aki Liu's 2019 paper in Aging. What they showed is that compared to actual PCR-based telomere length, the DNA telomere length actually had a better association to disease compared to just telomere length in general. And Hastings followed up in 2022, where they surmised that the reason why that might be the case is because you're picking out CPGs that are associated with the epigenetic changes in the modulation of the gene expression patterns associated with telomere elongation and maintenance and all of that. And so we can look at that overall. However, when we are developing omic MH and any of these predictors, we're not specifically saying, okay, only pick CPGs that are associated with telomere maintenance or all that. We are essentially putting it in and letting the data decide. So there might be certain CPGs associated with telomere length maintenance. However, we didn't specifically categorize that these CPGs were telomere associated or not.
It doesn't matter on the model. Okay, good. I have one technical question. I used to use these Illumina chips 15 years ago, the 27K in my PhD. And I recall the protocol was fairly long, like 2-3 days. My question is, in the clinic, would it be possible to measure it in the same day and decide what drug the patients need to get, right? Can you comment on the advancement of these arrays and maybe how you see it's being used in a year or 5 years from now?
Yeah, I'll start, I guess. I think that for us, the really big benefit of these DNA methylation algorithms, where this is going, is just the scale of data that you can get from molecular biology at the level of cost. And so going back to one of our first slides, we do really believe that with just one single drop of blood on a blood spot card, that we can report out a multitude of different data points which can help guide clinical care. So obviously those will be methylation risk scores for disease, but they also will be diagnostic specific methods, as well as sort of environmental exposure methods, as well as most of your basic lab models. And if we can outpredict these biomarkers to disease with right around 64%, we imagine that we'll be able to take over a lot of that clinical testing. So we imagine that this could be a test used multiple times a year as screening and tracking that can be very low cost and really easily accessible by patients. We still do require blood. Saliva is not yet ready for a lot of these things, but I think that already we're having physicians guide this in their preventive care for a lot of different conditions, especially using those metabolites and proteins as indicators of status that might tell them about which pathways are most affected.
Thank you, Ryan. I mean, that's...
And in terms of like the same day assessment, that's a question for Illumina if that technology will be improved, because for us, I mean, we're still pretty rapid in terms of like around a 2-week turnaround time. But I mean, beyond that, it's that's on Illumina, I guess.
And there's some great point of care things coming out, but generally they require very few CPGs. And right now, most of our algorithms to hit the criteria we need on specificity, sensitivity, and technical reproducibility do require more than what we're able to do via those point of care methods.
Cool. Thank you very much. We are the hour. This was a very great talk. Thank you from the BES to join us today. And yeah, we all look forward to the preprint on bioRxiv and hope it's getting published soon, and wish you best of luck with this company. Really, I'm really excited about the case to bringing it to the clinic and help patients to get the right medicines. Thank you both.
Thank you all. Appreciate you having us.
Thank you for the platform.
Of use in the in-person event in December if you're in the Boston area. Thank you.
There.
Bye-bye.
Thank you all.
Bye.